Hello everyone, welcome to Johnson's Biology. We are going to take some Cornell notes today, and you should be on Cornell notes page 23, so go ahead and put a 23 up there, okay? If you do not have a Cornell note sheet with you and you are at home, please just go ahead and use regular notebook paper. That'll be okay. Just outline it just like in your regular Cornell notes. Today our topic is enzymes. Okay? And our essential question is, how do enzymes speed up chemical reactions in my body? Well, not really my body, but maybe everybody's body. Don't forget to put your name up there and your date. Alrighty, the other day in class we actually watched a video about how enzymes work, and I want you to remember about Pac-Man and about active sites and stuff like that. And here's a quick little overview so that you can see a picture of how an enzyme works. This is an overview of an enzyme. An enzyme is a chemical in your body. It has a specific shape where there's an active site here. Some molecule, like a substrate, called a substrate, will actually fit in, and the enzyme will create a product. Now, enzymes can bring things together or they can break things apart. So please remember our video from the other day, and that will help you remember. So first of all, under our notes section, we'll go ahead and get started. Hi everybody, there's my hand. Okay, so first of all, enzymes are proteins. Okay, they are a type of protein. They are not fats, they're not lipids, they're not carbs, they're not nucleic acids. They are a form of protein. Okay, now what do they do? Now you may have written down a lot of these things on your notes from the video, but just in case you were absent for the video, let's get a couple things down that what they actually do. Okay, they speed up reactions in your cells. Okay, another thing, what they are, there's a synonym. A synonym for an enzyme is a catalyst. Okay, catalyst means to speed up. You can use this in the everyday language as well. For example, the catalyst that broke up the boyfriend and girlfriend was another hot girl. So that's what the definition of catalyst actually means. It's something that speeds up a reaction. Okay? They break molecules down or bring them together. Okay? They are crazy specific. So for every thing that you eat, there is a specific enzyme that breaks it down. Let's see. They can be destroyed or denatured. Denatured would mean it messes with the active site. So if we were looking at our active site again here, it would mean for some it had messed up that active site. So instead of having a big loop up, maybe it would be flat across. Remember our Pac-Man video where the Pac-Man's mouth was kind of messed up and it couldn't eat anything anymore. So they can be denatured by pH. So that would be putting them in too acidic of an environment or too basic of an environment, and temperature. Now, we're not talking about temperature like, hey, I have a fever, so my enzymes are messed up. No, they're actually talking about super high temperatures, like being cooked. So, for example, if we were to cook your hamster in the microwave and kill it, yes, none of his enzymes are going to work anymore, and you will not have any more hamster, and you'll eat hamster for dinner, I guess. That, or, I don't know, that's kind of gross, to tell you the truth. They can be recycled. Or reused. So one enzyme, after it's done with one reaction, can do the exact same reaction with a new substrate. They work specifically like a lock and key. So when we said they're specific up here, let's add a little bit to that. Like a lock and key. So for example, there's only one key to your house, the lock at your house. If your friend comes over and tries to put their keys in there, it's not going to work. Just like enzymes are incredibly specific. The enzyme lactase can only break down lactose. So let's go ahead and write that down. So for example, lactase can only break down lactose. Now lactose is the sugar in milk. So ACE indicates that this is an enzyme. OS indicates that this is a sugar. Okay, so this enzyme can only break down that sugar that is found in milk. People who are lactose intolerant, those poor people, they cannot have ice cream or any dairy product without taking this enzyme, or else they have serious tummy issues, and that's what all we'll say about that. Okay? If you don't have an enzyme, like if you didn't have lactase, your body for some reason just turned off the creation of that enzyme, which does happen. You don't really need milk after you've been born or after a couple years of life anyway. So if that enzyme gets turned off, then you become lactose intolerant. But there are serious diseases that can be caused by missing certain enzymes, and those can actually cause death. So they're very, very important for your body. Okay? Now, what I'd like you to do is just draw this diagram here, okay, on your notes, all right? You don't have to include what's a lock and key or induced fit. What I need you to do is draw the enzyme, draw the substrate, active site, draw them together, and then do the products. If you need to pause it so that you can do that, please go ahead and do that. So that should be right in your notes. Okay? Okay, so your picture is going to go here, but I'm not going to redraw a really cool picture. You can already have drawn that. Okay? So make sure in your picture that you have all the pieces and parts identified. Go back to the picture if you need to. There's a few things that we need to discuss. Active site is where some action is going to happen. See, active site, it's where action happens. Okay, the enzyme, this is what actually does the work. Okay, so it brings substrates together or breaks molecules apart, and we already said that up there, so we're not going to include that down here. Let's see, then we have a substrate. That's just a fancy word for what actually fits into the enzyme. So this is the molecule that is broken down or brought together. O-U, sorry, G-H-T, together. Okay, the enzyme substrate complex is just a fancy word for when they're actually stuck together. See, look at that. Enzyme here, substrate here. When they're stuck together, we're just going to put the word complex on the end to make it sound cool. So the enzyme substrate complex is when they're joined together. Okay? Then we've got our product. okay? And that's what actually comes off at the end. So sorry about that. What is released at the end. Okay? So that is what enzymes do. They're really, really cool. They are inside all of your cells and they help your body run. So anyway, that's the end of this lesson, and go ahead and do your Cornell questions. Go ahead and do a level 1, a level 2, and a level 3. Okay? And go ahead and do your summary. Please do a 2-sentence summary. And that is it. Have a great day. Let's turn this baby off.